Hello, everybody. My name is Ryan Phillips. I'm the CFO of the FOXG1 Research Foundation and father to a child suffering of FOXG1 syndrome. Like many of you probably watching this video, upon receiving a diagnosis of a loved one, in this case of FOXG1 syndrome, that's a genetic condition, you receive this code from your geneticist or neurologist. And at the time of receiving that code, it meant nothing to me. And until I was presented with a graphic that I want to share with you today. And this graphic really helped me understand more about the mutation that had occurred in my son's FOXG1 gene that resulted in the disease. And I hope that it will help you as well understand a little bit more of how genetic disease occurs. And then I'll also share a few ways that here at the FOXG1 Research Foundation, we're thinking about curing or improving the quality of life of children suffering from genetic conditions like FOXG1 syndrome. So this graphic was created by Dr. Adam Waite on the FOXG1 Research Foundation's Scientific Advisory Board. And it's made up of two images in one. The top half showing a healthy, or what's referred to as a wild type FOXG1 gene, and then the bottom half representing a gene that houses a mutation that resulted in FOXG1 syndrome. So the way to read this image is along the top, starting in the top left here, you have characters G's, C's, A's, and T's. And the order of these characters are coded. This is a DNA string. And it's the order of these characters are read or understood by the body to code for amino acids, which are shown in the center of the image here. And each three characters, so from the top left, GGA, each three characters represents a nucleotide or a codon. And each three, depending on the order that they're in, code for an amino acid. So GGA codes for GLU, GGG coded for GLY or glycine in this case, and so on. So every three characters, each nucleotide or codon codes for a amino acid. And then the amino acids are shown in the bottom here with a number on top of every fifth one. So the 170th amino acid here, 5 later, 175th, so that we know where we are in the gene when you're looking at an image like this. So then if you come to the bottom half of this image, that shows the FOXG1 gene in this case that harbors a disease-causing mutation. And this, when you compare it to the top half, you'll immediately identify that this C is missing from the wild type protein up here, or the wild type copy. And this omission of this single character resulted in what's called a frame shift. So all the characters after this deletion, this single character deletion, shifted into its place or forward one location. And the body continued to read these characters even though they're not in the correct order due to this deletion and shift of the character sequence. And the results of that is when you look at the amino acids, they're not the same as the top half. And that's because of the reordering of this DNA string that resulted from the frame shift. So you'll notice like the 170th amino acid in the healthy or wild type copy up top is LYS. In the mutated copy, it's ARG, and then ARG again instead of GLU, and it continues to be incorrect all the way until the 191st amino acid, where you see this red siren looking symbol. And that is where a stop codon was rendered, is what it's referred to as. And what that means is 3 characters in a row in this 190th location were in an order that the body didn't recognize. And it said, hey, I don't know what this is. Stop reading this genetic sequence. And so it doesn't read anything beyond the 191st amino acid where this stop codon was rendered. And so this mutation is written, and several people, caregivers or loved ones of individuals suffering from genetic conditions, have probably seen something like this. And this is written as C507del, which means that the 507th character, in this case, was deleted. And then furthermore, they give you a little more detail and they say PGLY169GLY-FS23. What that means is in the 169th amino acid, in the healthy copy is glycine, GLY. And in the mutated copy, that harbors a mutation. The 169th amino acid, which in the wild type or healthy copy of the gene is GLY, has a mutation in it. And as a result of that mutation, the amino acid actually remained GLY because GGA and GGC, in this case, both codes for glycine. So the amino acid in the 169th location that harbors the mutation stayed glycine. There are cases where, it will change the amino acid in the actual codon that the mutation occurs. In this case, it did not. So it's saying healthy copy has GLY there. In the mutated copy, there is a mutation in the 169th amino acid. It remained glycine or GLY. And that deletion, that mutation in the 169th location, resulted in a frame shift, which is what this FS represents. And then it says, and that frame shift resulted in an additional 23 legible amino acids until the stop codon was rendered. So from 169 to the 191, there are 23 amino acids that were sequenced in a way that the body still recognized what those amino acids are coded for until the stop codon appeared 23 amino acids later. So this is a much more detailed way to kind of write or explain what occurred. And that really helped us understand exactly what this image really helped us understand more what these at the time hieroglyphics were saying. And so I hope that helps everyone else understanding a little bit more. So the ways that we're thinking about correcting a disease causing mutation like this, there's really three main ways or buckets that you would lump therapeutics into. So first is correcting the mutation with something like gene editing, or CRISPR is a hot topic right now. So actually correcting the gene for the mutation, replacing this entire gene that is not performing the correct function with a healthy copy like a gene therapy, or overcompensating for the lack of protein in this case that's being created. So you can upregulate the protein that's being created from the healthy copy to overcompensate for the mutant copy. And so those are the three main categories and ways that we're thinking about developing therapeutics for FOXG1 syndrome. And there are several other genetic diseases that have successfully deployed strategies to do those things. So I hope this is helpful. Thanks for taking the time watching this video, and have a great rest of your day.